Have you ever just stopped and thought about one of the most mind-blowing mysteries in biology? How does a single tiny cell, with nothing but its DNA, somehow know how to build a whole complex creature? It's this incredible process of self-organization, and it turns out the secret is a conversation.
I mean, just look at this. In only a few days, you go from a handful of cells to this intricate little ball that's ready to become an embryo. And there's no blueprint, no architect telling them what to do. They're just talking to each other, coordinating every single step perfectly.
So, that's the billion-dollar question, right? How are they doing it? What language are they speaking? For decades, scientists have been trying to listen in on this cellular chatter, but it's proven to be really, really tough. Okay, so here's our game plan. We're going to look at this secret conversation and the big problem that's made it so hard to study, the problem of time. Then we'll dive into a new tool called TimeTalk, see what it's found, and explore how all this could lead from computer code to actual cures.
Right, so the biggest hurdle has always been time itself. You see, early development isn't a series of still photos. It's a continuous flowing process. It's a movie, not a photo album.
And that's exactly where our traditional tools hit a wall. Most of the software we have is built to analyze communication between cells that already know what they are, like a liver cell talking to a kidney cell. But in a super early embryo, the cells haven't decided yet. They're totally fluid, constantly changing. You can't just slap a label on them. So the old tools, they just can't keep up with the movie.
And this is where the hero of our story comes in. It's a new computational framework called TimeTalk. It was designed from the ground up to analyze this exact kind of fluid, time-based conversation. You can think of it like a universal translator, or a decoder ring for the embryo's secret language.
So how does it work? It's actually a brilliant three-step process. First, it just puts all the cells in chronological order, basically creating a timeline of development. Second, it identifies the key vocabulary, the signals the cells are sending out, and also the speakers, which are the master genes calling the shots. And third, and this is the really clever bit, it figures out cause and effect.
It does this using a nifty statistical idea called Granger causality. The concept is way simpler than it sounds. It just asks, does knowing what X is doing right now help me predict what Y is going to do in the future? The classic example is lightning and thunder. You see a flash of lightning now, and that makes you pretty darn sure you're going to hear thunder a few seconds later. TimeTalk uses that exact same logic for genes and signals.
So, with this powerful new decoder ring, what did the scientists actually discover? What is the embryo saying? Well, it turns out the findings are absolutely fascinating. For starters, TimeTalk identified 430 specific communication pathways that are absolutely critical for this early conversation. This is the core vocabulary, the essential dictionary that the embryo uses to build itself.
And what's more, this conversation isn't just random chatter. Now, this image might look complex, but the key takeaway is that the dialogue is organized into 6 distinct chapters. And each chapter is directed by a different group of master genes, guiding the embryo through the next big step in its development.
This chart really drives home just how good TimeTalk is at finding what matters. So out of all the genes in the genome, only about 9% are actually essential for survival. But when you look at the genes that TimeTalk flagged, a whopping 47% are essential. It's incredibly good at filtering out all the noise and zeroing in on the conversations that are critical for life.
Okay, so being able to listen in on an embryo is amazing science. But what can we actually do with it? Well, this is where that fundamental research starts to have real-world implications for the future of medicine.
So one of the very first things they did was use TimeTalk to compare a natural mouse embryo to a lab-grown model called a blastoid, which is made from stem cells. Now, these blastoids are super important because they let us study development without using real embryos. But the big question has always been, are they actually a good substitute?
And TimeTalk delivered a huge answer. It found that yes, the blastoids share the same core communication pathways. They are, for all intents and purposes, speaking the same language. This is a massive deal because it helps validate these models as a powerful and ethical tool for studying everything from development to new drugs.
And this just blows the doors open for future applications. If we understand the normal conversation, we can start to figure out what goes wrong in developmental disorders. We could potentially improve fertility treatments, learn how to build lab-grown organs, and maybe even unlock brand new kinds of regenerative medicine.
So this breakthrough, it's not just a new tool. It's a completely new way of thinking about how life organizes itself. It really is like we've finally learned to read the first chapter in the book of life.
And the secret it's telling us is that development happens in waves. You get one set of genes turning on, that triggers a wave of signals between cells, and those signals then turn on the next set of genes. It's just this beautiful, self-sustaining cascade of information passing from one cell to the next.
But of course, science never stops. These are incredible insights from a computer model, and the next step is for scientists to confirm them in the lab. And we have to remember, this is just one way cells talk. There are probably a whole lot of other conversations happening that we're still just beginning to tune into.
But this all leaves us with a pretty provocative thought to end on. For the first time, we aren't just watching development happen. We're starting to understand the language and the logic behind it. So, if we can truly read the script that life uses to write itself, could we, one day, learn to rewrite it, to fix the typos that lead to disease, and maybe, just maybe, write a few new sentences of our own?